My name is Mark Markiewicz from the National Institute on Aging, and today with me is Dr. Molly Waxter, who is heart and mind behind this Walls lecture. It is our pleasure to introduce our speaker for today's Wednesday afternoon lecture, Dr. Jonathan Yoni Kipnis. Dr. Kipnis is Alan A. and Edith L. Wolf Distinguished Professor of Pathology and Immunology at Washington University in St. Louis. He is also Professor of Neurology, Neuroscience, and Neurosurgery and Director of the Center for Brain Immunology and Glia at the Washington University School of Medicine. Before I tell you about, more about Yoni, you should know that if you are attending the lecture for CME credits, the code is 57686. 57686. If you are watching online and would like to submit a question, please use the Send Live feedback button, and your question will be relayed to the speaker at the end of the lecture. We are delighted to have Dr. Kipnis here today to talk to us about his studies on interaction between immune system and nervous system. His lab discovered a lymphatic vessel in tissue surrounding brain. That work brought a fresh perspective to research on neurodegeneration such as Alzheimer disease, multiple sclerosis, psychiatric disorders such as anxiety, neurodevelopmental disorders such as autism and Rett syndrome. Dr. Kipnis' lab studies the complex relationship between lymphatic system, glymphatic system, and transport across blood-brain barrier. Dr. Kipnis' group is also investigating the role of immune cells and molecules in brain function. He's working toward mapping the immune receptors repertoire of the brain, and also he's trying to understand how cytokines impact behavior. His lab studies neuroimmune interaction in the context of spinal cord injury, and by unraveling the cellular and molecular mechanisms, we hope to move forward toward novel therapeutics that employ the healing power of the immune system. Dr. Kipnis has been a member of the National Academy of Medicine since 2022. He received numerous NIH grants, including NIH Pioneer Award and NIA Merit Award. He has been studying interaction in the context of health, neuroimmune interaction, in the context of health and disease since his elementary school, almost, since his graduate school, when he discovered the role of naturally occurring T cells in the immune response to central nervous system injury. For his doctoral studies, Yoni received Distinguished Prize for Excellence from the Israeli Parliament. Dr. Kipnis earned a Bachelor of Science in Biology from Tel Aviv University and PhD in Neuroimmunology from the Weizmann Institute of Science. And he was a faculty member at the University of Virginia for 13 years before moving to Washington University in St. Louis in 2022. Please join me in welcoming Yoni, Dr. Kipnis, to speak about navigating uncharted neuroimmune waters.
Well, thank you very much, Mark, for this introduction. Thank you, Mark and Molly, for inviting me here. It's really a pleasure, and it's really fun to be here. It feels almost like home. I know so many people here and good friends. So it's really, really fun. I don't see you at all. I have these 4 sources of light into my eyes, and it's not fun at all, but I guess it's a requirement, so I'll have to go with it. So I also was told that this is like a big lecture, so I should do more. It's a very unusual. Normally I present introduction and then I show 1 or 2 stories which are unpublished and try to go in depth, and today it will be basically a little bit of a bunch of different stories. So hopefully if you lose yourself at some point, don't worry, I'm probably lost myself there as well. So I'll start the lecture with this quote. It says, it is what we know already that often prevents us from learning. I think it's true for every field. Think about your field for a second, and you will agree with me, hopefully. It's really very, very true for the field of neuroimmunology, anatomy of the brain and head, and the flow of CSF, and all these old concepts which people thought they figured it all out in the 80s with the use of EM, and none of them want to actually things to be any different than what they proposed back then. But things move forward, and I mean, so are we. So, of course, if you were to buy a house and it would look like this, you probably would not want that particular house. You probably will want your piping to look like this. And I think it's also true for all our tissues. And our tissues, all of our tissues have two types of pipes: the blood vessels, that I'm sure everybody in this room knows at least something about. And the second network of vessels are the lymphatic vessels, which I think many in the room don't know and don't care about at all, right? But these are very important vessels as well. So blood vessels are maybe similar to our water pipes at home, and the lymphatic vessels are similar to our sewage pipes at home. So blood vessels will bring all the goodies into the tissue, oxygen, glucose, nutrients, and the lymphatic vessels will remove all the waste products from the tissue out into the periphery. Before it gets detoxified, it goes through the lymph nodes. And the lymph nodes, for those who don't know, are the centers where the immune cells are sitting and literally going through the trash coming from the tissues. Just think your neighbors go through your trash cans. Right, so within basically 2-3 weeks, they will know all about you. And if they see suddenly one day something really weird in your trash can, I don't know, let your imagination flow. I don't know, maybe blood-soaked towels. They probably will call police, right? So same thing with the immune system. If they see that something abnormal comes to that tissue, which should not be coming from normal, healthy tissue, they would go, call the police, they would just go and explore what's happening in that tissue. And that's a kind of a very, very simplistic way to explain immune surveillance of organs, and that's what the immune system does. So the notion was that the second part, the lymphatic vessels, the lymphatic system, and the immune surveillance does not exist for the brain. So our most important organ, basically, was left out of immune surveillance, which is our most important surveillance and maintenance tissue in the body. Now, brain is, of course, extremely busy and extremely metabolically produces a lot of metabolic waste organ. And so how does the brain cleanse itself? And because there is really no lymphatic vessels within the brain tissue, at least there is no good evidence to suggest otherwise. So there is one very poorly done paper from NIH, maybe I shouldn't mention it, which is just wrong. But other than that, there is no lymphatic vessels in the brain that I think is agreeable on everybody. But the brain, again, like I said, is highly metabolic, and so metabolically active. And several years ago, Mike Nidegard came up with a very interesting idea. It was 2013 about. She came up with a very cool idea about how CSF actually serves, if you wish, like lymphatic function by being able to go through the brain tissue. So we, as you know, or those who don't know, our brains are swimming in cerebrospinal fluid. And at CSF, actually, as arteries are diving into the brain, we have a space in periarterial space. And CSF will dive together along with this periarterial space. Because of arterial pulsation, the CSF will be pushed into the parenchyma, will go through the brain tissue, and then it will collect all the metabolites on its way, and it will come out along the perivenual space. Here we made a very fancy movie, and I can show it to you. We paid a lot of money for it, so CSF goes in and then washes all the waste from the brain and comes out along the perivenual space. I mean, that's kind of it's again, very simplistic understanding of the lymphatic system, but I think it's probably, in reality, it's probably something that is not very, very far away from that movie if you're looking at particular brain region. And of course, the question is, okay, where all this junk goes then? How do you clean that real thing from also not coming back into the brain and not create kind of brain swamp? And so we, totally independently of this lymphatic story, we were very interested in brain meninges. For those, again, who don't know, these are the three-layered tissue that enwraps the brain and spinal cord from atop, right? So our heads don't just have the brain in it. It's also, there's a layer between the head and the skull called the meninges, with the P attached to the brain, dura attached to the skull, there's arachnoid underneath dura, and then the cerebrospinal fluid flows between the arachnoid and the pia mater. And that area is very, very rich in all kinds of immune cells. And what my lab was really mostly doing is studying the function of different immune cells in the dura, and they're derived cytokines and how they affect brain function, with a particular interest in how things are changing in aging and in different age-related diseases like Alzheimer's, and how that may play a role in those conditions. And actually, that was my bread and butter of my lab. That was my first R01, which is now actually in merit phase, which is very exciting. And that's how I got kind of into NIA in the very, very beginning. But I will not be talking about this today. I will mention it at the end a little bit, the news study that just came out. But I just wanted to tell you that we were very excited about this meningeal immunity. And we were asking ourselves, but how these immune cells are coming and going into the meninges? And so my postdoc back then was Antoine Louveau, and what he did, and she's now a PI at Cleveland Clinic. And so what Antoine did, he established this whole mount of the mouse dura. See, he peels the dura mater, the outmost layer, and he puts it as a 2D basically on a slide. And here the labeling is, these are in red are blood vessels, and you can see in the middle this Mercedes sign, or peace sign, whatever you prefer. So these are very thick structures. They call the sinuses, and so they drain the vascular, they drain the cranial, basically brain venous blood out into the periphery. Very important structures. And so along these structures, Antoine did staining with marker for lymphatic endothelial cell. Don't ask me why, it's a long story, and I will not be telling this to you now. But he actually detected that there is some staining there, which looked like vascular. Now, in the eye, we have a vessel that drains the eye. It's called Schlemm canal, but it's not made of lymphatic endothelial cells. But here, actually, we have a real bona fide lymphatic endothelial cells that express all the right markers. And so we ask ourselves, okay, is this functional? Can it function will it actually drain CNS or CSF? So here is the live mouse with the window in a skull. We inject the mouse with green tracer intravenously and with the red tracer into the CSF. And we're watching and we see that there is a green tracer from the blood inside the sinus, and the tracer from CSF is within this one little tiny vessel, which now if we turn the mouse upside down and we open the neck, we will see this red dye from CSF will continue to flow along this lymphatic vessel into the draining lymph node. Now, really nice. I love the mouse because they only have one lymph node on each side, and then there's two other ones which are superficial, but that's all. In humans, we have, I think, over 100 on each side of the neck, so makes it much less fun, but still very important, of course, to understand how things are working. So after we published our paper, there was a series of labs that were showing very similar results. At first we were told that we were all wrong and it's all irrelevant, but that's okay. We didn't last for too long. Karel Italo published a paper saying exact same thing that we showed. Then there was actually 3 papers from 3 continents. This was European. This was American from NIH, from Brent Weinstein's lab. Really beautiful story. And then there was a paper from Australia. All 3 showed the presence of these vessels in fish. So this was now evolutionarily conserved structure, also present in rats. And then again, NIH here, Danny Reich, with a little bit support from us, but really not much, even though he gave us authorship on the paper, which was very nice of him, showed the vessels exist also in non-human primates and in humans. And I will quote Danny. He said that it took him 2 days to find the vessels and then 2 years to convince himself that this is real. Am I correct with my quoting here? So I guess vessels do exist in humans. They exist even in fish, so they're probably real, and so we can understand what they're doing. Before then, I know many of you might have heard already this, but I would like to show you a brief history of CNS, of CNS lymphatics. There was an emperor of Habsburg Empire, Joseph II, really believed in education. And so, but because back then there was no way to preserve human organs, so he commissioned this very famous Italian physician, Paolo Mascagni, and together with his artist, Clemente Michelangelo Susini, they were basically making replicas of human organs and human parts of human body using wax. Precision was absolutely incredible. So this is a human brain and a full human head. Again, it's all from the wax. I mean, back then they were able to do this from marble, so wax probably was the very easy material to do these amazing things from. All this pasta that you can see here coming from the brain, Mascagni claimed to be lymphatics coming from the brain. I mean, based on Danny's work, we know that even though there is lymphatics there, it's not as intense as Mascagni was claiming. He actually got really criticized back then by whatever back then was called Papier. So really his contemporaries really killed that whole thing, and He died basically in infamously. Nobody even knows where he's buried, unfortunately. But I guess there was some truth to that. So I don't know whether he was either exaggerating or really was very unlucky or very lucky to work with a really abnormal, hyperlymphatic person, right, in this particular case. So in 2003, there was a paper in Lancet which summarized Mascagni's work. And it says here that today there are 867 models on display in the museum. Museum is Josephina Museum in Vienna. If you happen to be there, must visit. It's really beautiful. And it says that complexity and accuracy of models reveal the scientific excellence of their creators and their knowledge of gross anatomy, which is still accurate. However, 4 models stand out for their obvious anatomical misconceptions. Case number 1, lymphatic system on brain surface as anatomical misconception. And it's 2003, so I'm in the midst of towards the end of my PhD, so I'm learning based on that misconception, right? There's no lymphatics in the brain. And so here it says that the lymphatic system was the focus of Paolo Mascagni's scientific interest. So Mascagni was probably so impressed with the lymphatic system that he saw lymph vessels even where they didn't exist in the brain. And then it says the lack of lymphatic circulation in the brain was first described in detail in the late 19th century. How to describe a lack of something in detail? And you're being first in that. So it's a big series. How many other things are described in details to not to exist? And then basically we live in that misconception. Anyway, funny enough, we published later on some commentary saying that Mascagni was right, but we still have no clue where he's buried. So I do also hope that he's the right person to become infamous studying meningeal lymphatics. Okay, it was a joke, but I guess it wasn't very funny. Anyway, so my lab focused on lymphatic vessels really in 2 conditions. One is, like I said, we were very interested in aging and conditions of protein, of debris accumulation, so amyloidosis here. And of course, the one that is very close to our hearts is the neuroinflammatory disease, which is an animal model for multiple sclerosis. And so we focus in my lab on these 2. But actually, when we just made the discovery of lymphatics, I was interviewed by NPR, and they asked me, what is your dream about these vessels? And I said, I just dreamed that a bunch of labs are studying this, and now thinking maybe I should have something more substantial, because actually that dream came true. Because a bunch of labs are working on lymphatics, which is really fascinating. This is a very recent paper from Kathleen Caron's lab on the role of CGRP signaling on headaches that works through meningeal lymphatic vessels. Work on Parkinson's disease in patients. There is this paper on brain injury and the paper on hemorrhagic stroke. Again, all with connection with the importance of this lymphatic in these different pathologies. Even the response to stress that is dimorphic in different sexes in mice. This work on brain tumors and lymphatic, how they govern the immune response against brain tumors. And there was a couple of papers on the cognitive deficits associated with craniosynostosis, which are also linked to lymphatic dysfunction because of dysfunctional, kind of abnormal skull formation. And this is just really a handful of papers that I had on my computer, and I'm just lazy to put, maybe I can put probably another 100 different papers here of the role of these vessels in different diseases, so it's fun. So like I told you, I paid a lot of money for this movie, so I'll be playing it today a lot, and it justifies the costs. And I structured my lecture around this movie, so I'll show you first where this junk goes out, and as you can guess, it will go into lymphatic system, so we'll talk about this. Then I'll show you some interesting mechanisms, immune mechanisms, which govern the flow along the veins, and then the flow through the brain parenchyma. I'll tell you quickly why we think it's important and why I really care about this, and that will be the story. So if you thought I am speaking fast until now, be prepared, because now I'll start really speaking fast. I have a bunch of slides to cover. Anyway, so after this dirty CSF gets out of the brain, it has to reach somehow lymphatic vessels, which is basically the brain's sink, or if you wish, the brain's toilet, right? So that's where it needs to be all flushed out. Now, how do you reach the toilet if the area, if CSF is here, it goes to the brain, how do you get to that layer where you have to cross arachnoid? And if you read all the textbooks, it will tell you that arachnoid is placed there to prevent CSF access to the dura. So arachnoid should be a perfect barrier which doesn't allow CSF to actually enter the dura. So how does it happen? We don't know. The fact that it's happening not only in mice, it also happens in humans, and this is a paper from the Norwegian group that shows exactly what we're showing. They inject tracers in patients into the cisterna magna, and they see the tracer appears in the dura. So remember that if you're going into textbooks, and maybe there's clinicians here in the room who will now start throwing at me rotten tomatoes, which is fine, I'm used to it. So the textbook material tells you that arachnoid, this intermediate structure in the meninges, has those unique structures called arachnoid granulations or arachnoid villi. And they have these little holes here, and they are sticking all the way into the sinuses, into these venous structures, and pushing CSF directly there. And that's how you guys, I see you in a white coat, I assume you're a clinician, that's how clinicians are learning it, correct? Yeah. So, I was always bothered by this idea because, first of all, many animals don't have arachnoid granulations, and they still have CSF that is being produced. How is cleaning there? Our babies don't have granulations until they become much older. And also, how do you seal bunch of, if they're really sticking into the sinus, how do you seal that interface to prevent the blood from outflowing? It makes no sense. Anyway, so if you really look carefully into the anatomy, and which Rupal Mehta from Rice University did very carefully, I think it was last year or 2 years ago, and she showed that really arachnoid granulations are not going all the way into the sinus, they are coming into the dura, which very much supports our findings in mice and also this Norwegian findings in rats, in humans, sorry. We inject traces in the CSF, they're appearing in the dura, and then from there we think they're being taken by lymphatic vessels, which are in the dura mater. So, but how does CSF get into the dura? Because it has to cross a barrier which is supposed to be impermeable. And so Leon Smythe, who officially today is his last day in the lab as a postdoc, and he will travel through US and will start his position at Monash University in Australia in July 1st. So Leon, actually, this is our own EM of arachnoid, and you see the arachnoid barrier cells. They create this absolutely perfect barrier. So how do you cross it? And they're also very, very poor in transcytosis, so it cannot be really transcellular movement over the flow. Something has to be there. We inject different kinds of tracers, and they all arrive into the dura at the very same time. So transcytosis is not going to explain it. Something else is happening. There are some passages that are moving CSF into the brain. So I'm now delving a little bit into more anatomy that maybe probably any of you here wants to hear, but I have to explain it. So this is actually a quite ugly prep of a whole head clearance. It's ugly because, as you can see, the skull did not shrink as much as the brain did, so it created this very artificial, huge subarachnoid space, but it's very nice for demonstration because you can see these vessels here, these are veins that are bridging veins, or they're called cortical veins sometimes, and they're the ones bringing blood from the brain into the sinuses. And so they go through the subarachnoid space, all the way through all the layers of the meninges to reach into the dura. And so I'll do it in cartoon first, and I'll show you the images. So basically, this vein goes through all these things and then reaches into the dura to bring the blood. As it goes here through the arachnoid, there is basically like an opening or a tear, if you wish. So if you were to look at arachnoid from the top, you would see like holes. These are holes that are kind of coming, as you can see here. We call them, it's like cuffs, like a cuff on a shirt. And so the CSF, while the blood flows within, let's say, my hand, the CSF will flow within my sleeve. And so CSF kind of flows in the sleeve, and then it ends up in the dura. And then here it could be uptaken by immune cells, but also the lymphatic system is right there, and then whatever is left will just get out of the lymphatics. So this is how the structure looks in real life. So the green here is this arachnoid cuff, arachnoid structure that kind of goes along the vein, and within it, that's where CSF flows. And in mice, we showed that CSF goes there both ways, from the dura into the brain and from the brain into the dura. And then again, the cool thing about the structure also, this is where a blood-brain barrier ends or starts, depends how you like to call it. So blood-brain barrier is a very unique structure of endothelial cells in the brain, and so exactly at the end of that cuff of the sleeve, that's the end also of markers of blood-brain barrier in a normal vessel, peripheral vessel starts, which only tells you how important that area is to control, because now peripheral molecules can come out and then they can diffuse through this sleeve all the way into the brain. So lots of interesting control happening here, and that's some of the new works which we're now working on. So I told you in mice we showed both ways, up and down, but in collaboration with Danny Reich again, with Sirat, who is actually he's a postdoc in the lab, and I think he's here in the audience as well. What they showed very nicely that in humans, actually, they showed the movement of the tracer from the dura into the CSF, again, through those bridging veins. And so I'm not opposed to work in mice just for the sake of a mouse, but of course it's very fun when our mouse work translates to really human anatomy and human biology, so it's very exciting. And so here we really have amazing collaboration with Danny, which we are hopefully continuing with some other things which I'll mention in the future. So I'm not questioning then that the blood, so in the field of Alzheimer's, the current stage of the field is that, amyloid beta is being removed across endothelial cells of the brain, across blood-brain barrier. And it used to be like the only way, and we're not saying it's not right, we're saying there are other ways. And the lymphatic route is also another route to remove amyloid from the brain. And we actually showed that, and I will do it very quickly. If we're taking a mouse which is prone to amyloidosis, and we are killing lymphatic vessels in that mouse, we start seeing, first of all, amyloidosis now appearing in the dura mater, but most importantly, inside the brain, now the plaques are more of them and they are larger. So really, if you remove that route of excision, or that route of waste removal, it actually builds up, it ends up in building up more amyloid waste inside the brain. And we also showed that if we enhance those lymphatic routes, for example, and we combine them with anti-amyloid therapy, then we actually get a synergistic effect. So there was also a beautiful paper recently from a group in Mayo Clinic, and they showed that some of the Alzheimer's markers will appear in the lymph node, draining lymph nodes, cervical lymph nodes. They will be much higher than in peripheral blood. They also will be more predictive of disease development. So again, supporting this lymphatic route of removal in Alzheimer's patients than necessarily the route into the blood vessels. And so if that's true, then our sleeves, our points, which we believe are the exit point from the brain, should show some degree of clogging. And indeed, when we're taking a mouse just prone for amyloidosis, this is actually unpublished data, we're seeing that these areas are really almost like clogged by amyloid here, as you can see in yellow. And we collaborate with Danny on this as well, but we have now very nice first imaging from humans in collaboration with Rupal Mehra that shows that these ACE points, what we call, are also packed with amyloid in Alzheimer's patients, which is really exciting and actually supports this idea that some of the waste removal is happening across that route. Anyway, so this is for the way out, but now what about the way in terms of movement inside the brain parenchyma? So Mike and Hildegard showed that vascular pulsation, arterial pulsation, is very important to propagate the flow along the vessel and also inside the vessel. So Antoine Driot used to be a postdoc in the lab. He's now PI in Paris Cité in Paris. Was really interested in this function of the vessels. And if many of you here know about microglia, so if you label for microglia, you will find this beautiful labeling throughout the whole brain. But there is another myeloid cell which sits inside the brain, which is less probably known, definitely less known to neuroscientists. And these are the perivascular macrophages, perivascular to meningeal macrophages. So you can see that every vasculature that goes into the brain or out of the brain is covered by these cells here in teal. And so these cells are very interesting because if you look at the blood-brain barrier kind of niche, you will see, so from inside, of course, we have the endothelial cells of the blood vessels. On the outside, we have the glia limitans. And then inside, of course, we have the smooth muscle, we have the pericytes, but we also have macrophages, which are sitting right here in this color, which is purple. And the CSF kind of flows through. So basically, these macrophages are constantly sampling what is coming, what is happening with the CSF. And indeed, if you put a tracer in the CSF, then macrophages will be beautifully labeled by the tracer because they are phagocytes and they're constantly sampling and they're in touch with CSF. And also inside the brain. So we had this Antoine had this very cool idea. He said, they take up a space within the perivascular space. If we remove those cells, we will increase the space, and maybe more CSF will be able to go through, and maybe we'll improve the amount of CSF movement, especially in old mice when we have this problem. So I said, okay, that's a cool idea, let's do it. So what he did here is he injected this mice with obviously bubbles of liposomes, bubbles of fat loaded with toxic material, clodronate, Macrophages, again, because they uptake whatever is in the CSF, they will swallow it, and then the material will burst inside and will kill the cell. And we actually can get a very nice, about 60–70% of cells will be eliminated. So you can see there is much less of them. So we asked ourselves now what's happening with CSF flow in these mice. And what we found is was very kind of stunning, because we actually found the opposite. There was less CSF going around the brain, less CSF going into the brain. It's always fun to get results which is opposite to what you expected, because unless you switch the groups, it's a cool finding which you didn't expect, so it's also probably true. And we decided, okay, let's try and understand what's happening here. So what Antoine did, it was funny because we realized that there is no real good method to measure CSF flow in a live mouse. And what Antoine did, he developed such assays. So this is a mouse, for those who can recognize, this is the eye and this is the ear, and now you see the mouse, right, in the screen. And so what Antoine does, he lifts that piece of skin and a muscle which covers the beginning of middle cerebral artery. So we inject tracers into cisterna magna. It will go underneath the brain and it will follow the MCA and then will dive inside the brain. So we're looking at the beginning of middle cerebral artery, and you can see upon injection, that's the tree of MCA is being filled with the tracer. And now we can compare the normal mice and the mice that have no macrophages, perivascular macrophages. And the difference is quite obvious, even though here we quantified it, but the difference was really robust. It's very fun because when you have such a big difference, you can really study and understand what's really happening there, at least partially understand the mechanisms. So long story short, it's all published, so I'll just do it very quickly. So long story short is when we put mice in a high CO2 environment and they need to vasodilate way more, we realized that the vessels that did not have macrophages around them could not dilate as well as normal healthy vessels. So remember, macrophages are like tiny chimney sweeps. They're sitting there and they're constantly eating the debris which are the other cells deposit, right? So they're constantly cleaning extracellular matrix proteins. So if you remove macrophages, you start seeing built up of ECM around the vessels. And we believe that this ECM is what actually prevents vessels to fully dilate, because it's just built up and you have a gunk along the vessel. Now, in old mice, we knew that old mice have very substantial problem with CSF flow, and here we're showing it with live imaging. You can see this is a normal young mouse, 2 months or 3 months old, and the 24-month-old mice have very, very delayed CSF flow. It was also known that old mice have accumulation of extracellular matrix along the vasculature in the brain. It's not new. And we also realized that the types of perivascular macrophages are changing completely in old mice. So here now we're taking an old mouse and we're giving them MCSF infusion. It's a growth factor for these perivascular macrophages. And within a couple of hours, maybe 6–7 hours, we get reduction in accumulation of perivascular ECM, and we get suddenly now more CSF can go through the vasculature. And also inside the brain. So it was really a dramatic effect. It did not last for too long. It was quite short. It was only lasting for 24 hours, and then everything went back to normal. So, but again, gave us a glimpse of hope that maybe we can do something. And so we're now we have several collaborations going on where we're trying to make this effect to go much longer, and so to really get some substantial benefits to the brain. But the other ways, I always tell my kids, the beauty of being a scientist, you can destroy things and it's still valid and it's very exciting. So if we cannot fix them in perpetuity, we can maybe destroy them. It's much easier. So we're again going to our amyloidosis mouse model, and if we're destroying those macrophages for now quite long term, for 1 month, we see again accumulation or more accumulation of amyloid plaques inside the brain. Again, we are preventing that CSF flow through the brain, which leads to accumulation of plaques within the brain. And finally, what allows the flow of the CSF through the brain parenchyma? I know even in this slide, even though we try to avoid this, you see there's lots of black space there. But sometimes when you look at these commercials from companies about how drugs work, you see this empty space. There is one neuron, and then the molecule arrives, and then the other one arrives, and it's like super beautiful. And you realize, no, it's not how a brain looks like. It's a very, very condensed extracellular matrix, with lots of cells, lots of extracellular molecules. It's a super condensed tissue. So how does flow go through that very dense parenchyma? And that was something that Li Fang, Qian Xie, was postdoc in the lab, was very interested in understanding. And he actually came from Sleep Lab. He just opened his lab at UPenn, really brilliant guy. And so he asked what propagates the flow through the dense brain parenchyma. And again, he was coming from sleep lab, so he said, no, as a field, and I'm not a sleep biologist, I'm not even sure I'm any kind of, I'm a neuroimmunologist. So. But in sleep world, I mean, one of the big questions is why do we sleep? And I think one of the new and exciting theories for sleep is that we actually, when we sleep, we cleanse our brain more efficiently. And we know that some of the, for example, of our tracer experiments are working way more efficiently in a sleeping mouse than a wakeful mouse. There's no question about it. So I will go through this. This is a very complicated paper, and it's published last year. I will do it very, very quickly. I'll just summarize the whole thing because I think the concept is really exciting. So you know that when we sleep, we emit waves, and we can actually record those waves, and by the type of the waves you emit, my ring, and I'm sure many of you have this Aura ring, will tell you what phase of sleep you are, right? Deep sleep or REM sleep and whatnot. And so, so why do we have those? What is the function of these macroscopic waves during sleep? And so Lifang suggested that, so basically if you think when neurons are firing, during sleep what happens is neurons are firing together, this synchronized neural activity, which generates quite substantial surges of ion flow. And so this is the waves that we're recording. And so in deep sleep, we have very slow waves with very high magnitude. And he suggested that maybe this power of basically pushing all these ions into the interstitial space will be sufficient to actually also carry the water molecules with them, which will also carry the metabolites within it. So again, and so here we're recording, he did recording, this is a 32-channel recording, so we record the muscles in the neck, we record the activity of the cortex, and then 28 channels are recording in the hippocampus on one side, and you can see an animal that is sleeping, you see appearance of this macro waves and that you can see synchronized neural activity during sleep. So we can silence neural activity in these animals using chemogenetic approach here. So we express modified receptor of glycine. I mean, again, it will come in the next slide. And so when we give the mice ligand that binds to this receptor, we interfere with neural function and silencing neural activity. So you can see here, like all the waves are being suppressed in these mice, but overall in the hippocampus. But if you look at overall global brain, there is no effect. So we're really affecting a very, very small area of the hippocampus. And now we're asking ourselves, will this be sufficient to affect the amount of CSF flow through that brain region? And so here is the experiment. We're expressing the PSAMs in the one side of the hippocampus, or it's either control or just GFP, or PSAM itself or GFP. Then after 3 weeks, we inject them with the drug, which within a couple of hours will silence neural activity. Then we inject them with a tracer, and we either leave a mouse untouched, and that's during daytime, so mice will just fall asleep. And we monitor this, of course. Or we will bother them slightly. We'll just have this rod that will go through the cage every 20 seconds, and we'll make sure that mouse is gently kind of bothered and not being asleep for another 1.5 hours, and then we take the brain out. And what we're looking how much flow, how much CSF goes through that particular area of the brain with and without neural activity. And so what I'm going to show you here is that when we suppress neural activity, we see that there is, first of all, indeed in sleeping mouse, we have more flow goes through that area for the whole brain, but also through hippocampus. So again, when mouse is sleeping, there is more traffic of CSF through the brain region. But when we suppress neural activity, we also reduce the amount of CSF going through that area without affecting anything around. Li Wei-Tsai has a beautiful story now about how another type of stimulation, 40 hertz, which is very, very fast waves, that actually affects amyloid removal from the whole brain. In our case, and also they activate one region but see effect on the whole brain. In our case, it seems to be a very, very local effect. We can also mimic this. We can actually induce neural activity by optogenetic stimulation, and we can increase the amount of flow that goes through the brain region. Again, it's a very complicated paper, but you should just read it. It's quite exciting if you are into that area. So then what basically Lifang proposed, don't worry, it's another movie. What Lifang actually proposes during daytime, our neurons are active and they produce a lot of metabolites, right? And so during nighttime, when we know our brain is full of junk, neurons started to act more in synchrony, and then so they kind of fire together, and that creates those waves, which allows kind of removal of the waste or kind of movement of CSF more efficiently through that region. So in 92, Carla Schatz summarized the Hebbian hypothesis, And she said that neurons that fire together wire together. And so inspired by that, Li Fang suggested that neurons that fire together shower together. Okay, so that's basically that's the idea. And of course, the implications are quite exciting because of course, one of the major, again, I didn't bring any too much unpublished data here, but one of the major phenotypes of aged mice is that they don't really sleep very well. And we're actually now kind of forcing that clearance in aged mice, and we show improvement of sleep, which is super exciting because my goal, if I have one goal, is if I can just reduce my sleep to just 1 hour. So it's all about cleaning, right? So can you make your cleaning during night as efficient as it possibly can be in a very, very short time, and then just wake up and go back to your stuff? So that's a very exciting thing. Anyway. So if I'm invited here for a more smaller talk, I'll come and bring all these unpublished results. Anyway, so why do we care about all this? So what happens in the brain when lymphatics are really impaired? And so this is a work of KD in the lab, who is postdoc fellow. So again, very briefly, it just got published, so it's online, and I'm not going to elaborate too much on this. So KD, we knew that if you take young mice and you interfere with their lymphatic vessels, we will have all kinds of different cognitive dysfunctions. But really, we did not know the mechanism. We couldn't understand how come all this is happening. So first of all, KD did not believe any of this. He said, let me check first of all myself. And so this is a very simple, he did several essays in the paper, but this is like a very simple one where you introduce the mouse into Y maze in the water, and you close one arm so mouse can explore one arm. Then you open the second arm, and normal healthy mouse will go into the new arm more than in the old mice, into the old arm, because they're curious, and so they want to see what's in there. So they remember the old arm, and then now preferring the new arm. But mice that have lymphatic dysfunction, and in this paper we did 2 different ways of lymphatic dysfunction, they do not have that preference. So they're basically impaired in their novelty exploration behavior as well as some other behaviors. And then KD actually looked into medial prefrontal cortex. He was doing recording, physiological recording from medial prefrontal cortex. And while there was no effect on excitatory inputs, he showed a very obvious reduction in inhibitory inputs into the MPFC. The number of neurons which are sending those inputs was there's no problem with number of neurons. They were not dead. It's just the inputs were reduced. And so, we of course thought microglia could be the explanation. Indeed, if you take a mouse without microglia, these are fire mice, so they have mutation in promoter of MCSF receptor, and so the mouse is born. It has all the macrophages in the world, but doesn't have microglia. Quite amazing mouse. It's made by David Hume and Claire Pridon. And so in these mice, actually, really, if you ablate lymphatic vessels, we don't get that inhibitory synapse dysfunction. So really, microglia are playing a key role in this case in transmitting lymphatic dysfunction to synaptic dysfunction. But we thought, okay, so they're probably eating those synapses, as was proposed by Beth Stevens and many others. We did not see that. We see the eating, but there was no change in the amount of eating. What KD did detect is that there was upregulation in IL-6 interleukin-6 in the brains of these mice produced by microglia. And again, I'm now summarizing probably 4.5 years of work into really one slide. I'm doing huge disservice here to KD because he put really hard work into this paper. But again, it's published, so it will be easy to go and follow. Which he took, in this case, this what I'm showing you here is these are mice that have no IL-6 receptor on neurons. So we know that IL-6 is produced by microglia in response to lymphatic dysfunction, and they signal onto neurons. And so if we take this, we also abrogate this inhibitory synaptic dysfunction. So it's a microglial IL-6 which signals on neurons, which induces this synaptic physiology, which, as we discussed today with Lorna, there is lots of open question here about how really it's happening, what's happening within the cell. So aged mice, again, it's a pattern you're probably seeing, right? So in our lab, we're studying basic physiology, and then we are applying it to aging, and then we're looking what's going wrong in models of amyloidosis. That's kind of we're not classical aging labs because we're studying kind of what's happening in young mice and then how these things are going wrong in aging. So in aging, of course, we also see reduction in inhibitory inputs into the same MPFC region. And also what we know in aging is that lymphatic vessels are severely deteriorated in aged mice. I mean, that's also not new. We showed this before. Now, we don't know how to maybe improve synapses, but we definitely know how to improve lymphatic vessels. It's very easy. We actually infect mice with a virus that will express VEGF-C, a growth factor for lymphatic endothelial cells, and then lymphatic vessels in the border of the brain are just expanding very substantially. They now are no different from young mice. And we're now looking into synaptic activity, and we actually see increase in inhibitory synapses. Now, excitatory synapses were also reduced in old mice, but there was no effect here with lymphatic enhancement. Only inhibitory synapses were affected. Again, super exciting work. I mean, it kind of gives us a glimpse of understanding of molecular mechanism, but not full picture, of course. So we're now trying to understand a little bit deeper of what signals microglia receive from dysfunctional lymphatic to produce all these mediators, and of course how these mediators are really intracellularly affecting the synaptic inputs into neurons. And so finally, if you will allow me maybe 5 more minutes, is why do I care? Why do I care? Because I'm a neuroimmunologist. And with all my love to synaptic physiology and all this, I mean, this is all very nice and fantastic and flow of CSF, but really what I care is understanding how the immune system sees what's happening in the brain and how these processes are impaired, again, in different diseases and, of course, in aging. So what I showed you is we make clean CSF. It's going through the brain tissue, and it comes out as dirty CSF, carrying metabolites, but also carrying molecules, proteins, peptides from the brain itself. It will then go into the dura, as I showed you, and in the dura, then it will become the CNS lymph. So the dura part is very, very exciting because as the molecules kind of coming out into the dura, they're being uptaken by, I told you, there's lots of different immune cells in the dura. So they will be uptaken by these local, they're called APCs, antigen-presenting cells, and will be represented by patrolling cells. And then these cells, if there is recognition, they will start an inflammatory response, and if there is not, they will leave through nearby lymphatic vessel. So basically, if you wish, this is here presentation of the brain immune code. And we thought, okay, then probably then if there is a problem in the brain, then this immune code will change because you will start expressing new proteins and they will be presented. And so potentially this signature here, this barcode here probably will change way, way before disease really starts. And so we were very excited to understand what is being presented in the borders of the brain and how this is relevant for brain health. Again, very briefly, Mitch Kipnis was because he just left the lab, went back to med school, really brilliant MSCP student, MD PhD student in the lab. So in collaboration with actually Emilio Nanoe, who passed away 2 years ago, we started basically, we took all these border cells which present the peptides, we took these MHC molecules and we eluted peptides from them to understand what is being presented. And again, I'm doing it very quickly. It was just published a couple of months ago, so it's online. And what Mitch showed very nicely is that he found lots of interesting self-proteins, but one of them was a myelin basic protein, which is, as you know, a very dominant protein of myelin. But we found the peptides presented, which no existing algorithm predicted that they would be presented on MHC class 2 in black 6 mice. Now, interestingly enough, these peptides, for example, here, not only we found them in this strain, we found them in different strains of mice and also in humans. They were presented also in human subjects. So we asked, what are these peptides doing? They may have some unique regulatory maybe role. And so indeed, so if this is induction of disease in a mouse when we immunize them with MOG, so we immunize with another brain protein in the periphery, cells will go into the brain and will cause paralysis, the disease called EAE, animal model of multiple sclerosis. If we co-immunize with a SMOG and our endogenous peptide, we're actually seeing substantial reduction in severity of the disease. And if we modify the peptide, then it will lose its effect. So it's really something, this peptide is driving something very, very specific. And I will not kind of bother you with details, but what we showed is that this peptide induces endogenous cells, the T cells, of a very particular phenotype, the suppressor cells. And so they're constantly present there, and they're being recruited to the brain, and they suppress endogenous immune responses to really prevent kind of uncontrolled or very aberrant inflammation in the brain. Now, in disease conditions, what happens is that those protective peptides are disappearing. You can see here, I thought they would go up, but actually going down. And that potentially is another layer that allows more severe inflammation to go on. So we said, can we actually replenish? So if you think of a barcode again, it's like losing those some of the bars are getting kind of wiped out. And so we said, can we actually reinstate them? Can we bring them back? So we now packaged our peptide in those extracellular vesicles, and we're injecting them into the CSF. So now they will be uptaken by these brain APCs, and presented in the borders, and actually that suppresses the disease very, very nicely. So basically what we are suggesting is that the brain is constantly being washed, and the brain signature, the brain peptidome is being brought out into the dura, presented there, and is constantly surveyed by the immune cells. And that signature, when it changes, that's when the kind of immune response is changing. So normally we have this normal scanning of the brain immune barcode, but in disease conditions, like I showed you in EAE, in this case, the molecules, one of the major cells are missing, and so the immune response becomes abnormal. And instead of kind of suppressing the response, becomes more of an activator type. And so if we're looking into the aged mice, again, the signature of proteins is completely different. And also in AD, this is my favorite slide. So this is only 3-month-old mouse and 4-month-old mouse. And if you're looking inside, in an inside circle, this is the 5XFAD, mouse that are prone for amyloidosis. You can see it in yellow is the amount of the suppressive peptide that we found. And you can see within 4 months, it disappears completely. We cannot find it at all. So now the question is, what happens to AD if we reinstate the peptide? Why the peptide disappears? So what is happening there, and how immune cells are changing? Or maybe just because the flush of the brain is not good, and we don't get this peptide coming out, or some other mechanism. So lots of very exciting questions. And finally we ask ourselves, so can we maybe detect that immune barcode, immune signature of disease in patients? This is a very, very small prep. This is only 5 and 5 patients, AD and 5 PD. We now have a really nice amount of patients. We have now the samples for over 6 months already. We just bought a very fancy new mass spec, which is supposed to give us a much better reads on a much smaller sample. So we're just waiting for it to be fully running well so we can put all our samples through it. But even with a small sample, we already see there are some peptides which are only appearing in AD and others only appearing in PD. Actually, my student tells me that if I give him a sample, it's either AD or PD, he'll be able to tell me whether it's AD or PD, because some of these peptides are very, very only either or. So again, this is just kind of a dream that maybe one day we'll be able to do this kind of, I mean, it's a little bit more invasive than blood tests, it's CSF tests, but if we are able to get enough material from maybe CSF, this is all done from Dura. But potentially, maybe we could develop really exciting diagnostic tools to predict different diseases of the brain. I mean, again, this is a little bit of a wishful thinking, but I really hope one day it will become reality. So I showed the data of people and thanked them along the way as I was going. Many other people in the lab, data of whom I wasn't able to present. You know, of course, many collaborators at WashU and outside of WashU, many alumni, and many funding sources. You can see here. I really would like to maybe emphasize the NIA, because I mean they believed in my things at a very early in life, and when neuroimmunology wasn't really a fancy thing and nobody wanted to deal with this immune effects on the brain. So I was very lucky to get support from NIA, also Cure Alzheimer's Fund and other institutes here as well, many other sources. And I guess I'll stop here. Thank you.
It's not working. Oh, that's not working either. Cup your hands.
Yes. Okay.
Oh, it works.
So, one question. Did you check the myeloid cell compartment, and do you think that this change in peptide presentation could be related to the immune proteasome or to the type of myeloid cells which process the...
Yeah, brilliant question. So immune proteasome, I don't think so, because we're focused on MHC class 2. And so I think immune proteasome mostly controls what will be presented on MHC class 1. So we focus on MHC class 2. But in a very simple experiment, so first of all, if you take myeloid APC versus B cells, which are also APCs, and you load them with the whole spinal cord homogenate, or whole brain homogenate, and then you see what has been presented, then we cannot find our suppressive peptides presented by B cells. Never. You know, which of course kind of gives us very cool but potential ideas why anti-B cell therapy may be working in MS, because maybe you're removing all the APCs that cannot present inhibitory peptides. I mean, again, it's just a hypothesis, which is exciting, but we need to prove that it's the case. We also know that when you activate microglia or any other myeloid cells, you activate a bunch of different, and we show this in the paper, you activate a bunch of different proteases, which of course can cleave proteins different way, and then they will be different. We also don't know right here whether the cleavage happens in APC or in the cell of origin. So is it pre-packed that way, or is it being chopped by APCs? So, and we showed actually if you take oligocultures, MVP is coming from oligodendrocytes, and you treat them with different LPS, let's say with interferon gamma or some crazy activation, you will start seeing also the release within vesicles, they will contain different peptides.
Yeah.
So, I mean, this probably, it's a combination of probably lots of things.
So then, like, going back to this last point, and then I'm leaving. Like, when you have an infection or like a meningeal infection, like, then do you see also this switch and like a reduction of the regulatory or the suppression mechanism?
Yes, so what we think is happening, actually I was talking with Ken Murphy and I said, Ken, it's funny because our suppressor peptide, instead of going up, it's going down. And I said, yes, because your EAE model, because you induce a severe inflammation in the periphery, and so maybe the bodily tissues respond to, okay, if there is a problem, we know we need to maybe allow immune response to fight the infection. And so this could be very well that, that peripheral inflammation is being sensed by APCs around the brain, and now they don't present any more suppressive peptides to allow more efficient immune response against them.
Is it type 1 interferon dependent?
Yes, it is as well, yeah.
Let's go over here.
Yes.
Hi. So if you've shown that the peptides are MHC class 2 restricted, do you believe that Tregs are what's suppressing this inflammation? And if so,
oh, no, good question. Yeah.
Sequencing?
I kind of glanced through it. No, I didn't show you. No. So these are, when we immunize with our peptide, we do see some induction of Tregs, but very few. So we show that the best correlation is the non-classical suppressor cells. So these are FOXP3 negative, CTLA4 positive, IL-10 positive. So these are like, if you wish, like TR1 type of cells. And we show that if you inhibit IL-10 or TGF-beta, then you lose this inhibitory effect. So it's not classical Tregs, even though you do see them as well. And we now made tetramers against this MVP, our MVP, and so you can actually detect those endogenous cells. And normal, that's a beautiful thing, right? In normal, healthy, naive mouse, you see them in skull bone marrow, in the dura, and in deep cervical lymph nodes, and nowhere else. So they're really around the brain. And while majority of them are CTLA4 positive, FOXP3 negative, you also see some of them which are FOXP3 positive cells. So you do see some Tregs. Yeah.
Awesome, thanks.
Dr. Caspi?
That was fantastic. So MHC connection. If you postulate that the type of processing and presentation that occurs for the peptides that you see there has a major effect on disease and physiology, then you should see really strong MHC associations. So could you talk a little bit about
which you do with Alzheimer's, right? And MS. Am I correct? And I think a bunch of other diseases, but I could be wrong. I think MS, I think AD as well. AD, I think, is very strong for sure, yeah. Yeah, I don't know if I answered your question. So, I am not necessarily saying that these things are the driving force of the disease. So, but it's so it is very possible that this will just end up maybe being a diagnostic tool. So you see changes are happening. Maybe the outcome to these changes is not affecting progression of bunch of different diseases. I do think it affects progression of AD and definitely of neuroinflammatory conditions, but maybe some others it's not. But it's still, I do believe that when disease starts in the brain and you start making or producing some different proteins, again, if that flow thing is real, and if really the brain waste is being washed into through CSF into the dura, and the cells are presenting these peptides, then you will start seeing the change in that signature, which if you can detect and early enough, I mean, that could be, just by itself, could be a very, very exciting thing.
Certainly.
Over here, please.
Very nice talk. I guess on a similar note, since you showed that what age the CSF flow decreases and what fires together showers together. But then during early development, do you see unique patterns of CSF flows in particular?
Oh, yeah, very, very interesting question. So, one thing we are not doing in the lab is the developmental aspects of, so we don't do neurodevelopment at all.
Okay.
And there is a bunch of very exciting questions. For example, this brain meningeal lymphatics, for example. they develop after every other lymphatic bed. So, for example, a mouse is born, it has all the lymphatics, but it does not have meningeal lymphatics, and they appear around after about 3 weeks.
Oh, wow.
And actually, funny enough, they almost appear like the brain itself. There's a lot of them, and then there is a pruning, and there is less of them. And actually, one of my postdocs, Antoine Louveau, he actually is studying it, and I'm hearing that his paper may be coming out soon. So we will see what he will show. So yeah, I don't know what's happening in with CSF, patterns of CSF flow in early development.
Yeah,
but it's a very interesting question, of course. Yes.
Okay, yeah, especially with spontaneous activity.
Yeah, well, remember also that even microglia arrival, right? When microglia arrive into the brain, the arrival of the cells and their dispersal throughout the brain, first along the arteries and then along the brain tissue, it actually follows the patterns of the flow. So it's probably there way before. It probably kind of establishes itself. I don't even know when when Corbex starts to produce real CSF in mice. I should know. I don't know that. But these patterns of kind of what flow would look like, already you see it with appearance of cells in the brain.
Okay, thank you.
Yeah, pleasure.
Maybe 2 more questions, then we have a reception.
Okay.
Hi. I appreciate the mention of sleep as we start to learn that it's more of an active process. And I can imagine that brings its own challenges when developing your experimental designs. But my question is, how do you anticipate the tie with aging immunosenescence systemically, and also the role of the gut microbiota?
Oh, wow, this is a loaded question. I will say it's super complicated questions, right? We're also kind of trying to stay. So my neighbor 3 floors above me is Jeff Gordon. So every time we do a microbiota, we kind of try to make sure that we know what we're doing, and we have amazing neighbors for that. Actually, we just got IRB to do something with microbiota in Alzheimer's patients, which is very exciting. But again, I cannot talk about it right now. So it's just the beginning of the project. So definitely, all systems are cooperating and playing together, and I cannot really tell you exactly what's happening with this particular pathway, What I do know is that microbiota is changing, barriers in the brain are changing, lymphatic vessels in the aging are changing, amount of CSF that's produced is changing, amount of lymphatic vessel that functions is changing, and there's lots of interesting things. But I don't know if totally off topic maybe of the question, but I don't know if you've heard this new surgical approach in China that is now all over the news about this. They're doing this. They do anastomosis of lymphatic vessel in the neck to the veins in Alzheimer's patients, and they report some amazing results. Again, nothing was published, so we cannot really assess the details. But from what's being shown in media and what they can report in lectures, I mean, it's fascinating. So I hope it's true, but I don't know. I mean, we need to live and see. But I remember sleep. Remember also, while I do believe that in sleep the brain clearance is at its maximum capacity, I am not saying that there is no brain clearance during wakefulness. And I know there is a lot of arguments about this now between some groups. We always remain kind of neutrality, because I think that it's definitely happening during daytime, but it seems to be more enhanced during nighttime. Also, different types of waste, different topology of the brain may require different approaches to remove it. And so some may be removed more during daytime or in daytime and sleep time. Not sure if I answered your question, but I certainly answered somebody's question.
Last question, I think, right? Ine Belfer and CCH, thank you so much for the phenomenal story. So based on all these discoveries, what is your prediction about non-surgical, not invasive, maybe non-pharmacological manipulations of lymphatics and CSF flow in order to improve or prevent some of the aging disorders or other symptoms and diseases? So no,
I used to be at the University of Virginia, just 2 hours down the road, and once I was invited to give a talk in Blacksburg. I think it's not Virginia Tech, but the other place. It's not a MD school, but it's the other. Osteopathic medicine, yes. And somebody asked me, what do you think brain massage will do to lymphatics? And I said, I have no clue, but sign me up. And funny enough, actually, we just recently proposed a proposal to ARPA-H about not brain massage, but basically kind of almost manual therapy for lymphatics along the neck. to potentially improve the suction. It did not go very well, but I do believe that there is something there. And again, if you talk to manual therapists, they tell you these beautiful, amazing stories. So there is something there. I wish, you know, we need to get a little bit more down to a more granular mechanistic level. But
this is very promising. Also, we have a couple of studies that we funded for sound-based manipulations and for maybe acupuncture.
Are you Ina?
Yes.
Okay. Yeah, so you know our work then, which we're doing in part with.
We're very much we're watching what you're doing, and
you know, I always thought that standing on my head is a good thing. So I even bought this thing and I would stand up. But now when we through your funding, we actually saw that when you put the mice tilt down or tilt up, the flow of CSF is completely different. It's changing very, very much. And I'm not sure anymore I want to stand with my head down. So not to say that you guys should not do it, but I'm not sure. I don't know what it does to your CSF drainage out of the head.
Thank you so
yeah,
much.
thank you.
Okay,
thanks.
So quickly, the CME code 57686, and there's a reception outside, hard-earned reception. See you out there.